Hi, I'm Jack Fearey, and today I will be talking with you about the methodological considerations in mark-recapture of signature whistles and bottlenose dolphins. First, I would like to thank the ABC team for putting on this conference, as well as all of the support we got from Stellenbosch University. Firstly, I'd like to explain what signature whistles are, as it's the pillar of this project. A signature whistle is a unique stereotyped signal that each individual in a population will develop and use to broadcast its identity. Here are two examples of signature whistles from the population I'm studying. You can see these are tonal signals with a specific shape, and each individual will use this specific signal in order to broadcast its identity and to maintain group cohesion. Oftentimes, signature whistles are used to facilitate reunions between mothers and calves or between males in male dyads. And because acoustic communication is the preferred form of communication in water, as visual senses or olfactory senses are dampened, they make up a large proportion of the vocal repertoire of bottlenose dolphins. Historically, signature whistles were identified in captivity, whereby an individual was isolated in a tank, and the signal that was emitted the most would be determined as its signature whistle. Also, signature whistles were identified in wild populations during capture and release events in which an individual was captured, and again, the signal that it emitted the most was determined to be its signature whistle. However, more recently, a method to identify signature whistles in free-ranging populations was developed. This method is called SIG-ID, and it's a bout analysis approach, whereby you look for stereotyped whistles, and you need a minimum of 4, and these 4 whistles, 3 of the 4 whistles must occur within 10 seconds of each other. So here is an example of 5 stereotyped whistles in which they are produced within 10 seconds of each other, thereby qualifying this whistle as a signature whistle type. This presentation will focus on one chapter of my master's. However, I'd like to give you an overview of the project as a whole. So we want to use naturally occurring, uniquely identifiable acoustic signals in mark-recapture, spatial capture-recapture, and social network analyses, with the ultimate aim of developing an individually based monitoring system. So the different facets of this project include methodological considerations, so investigating array size and configuration of our recording devices, as well as looking at recording duty cycling in order to maximize and get the most accurate data set. Also, in the future, we'll be looking at vocal behavior and signature whistles and how these affect the various statistical analyses we will be using. So investigating the production of signature whistles in relation to group size and composition, as well as whistle matching and vocal accommodation, and then looking at the vocal production learning of signature whistles based on sex, association levels, and relatedness. And then the ultimate aim of this project is to do a home range acoustic survey of their entire home range, in which we use spatial capture-recapture analysis to investigate density and activity centers, as well as using signature whistles to invest in social network analyses. But again, today we're just going to be focusing on this one section of methodological consideration. The data for this study was collected using passive acoustic monitors. We used sound trap recording devices, which are autonomous recording units that can be moored in the water column for weeks to months. So we attach a sound trap or a hydrophone to a buoy and anchor system, and then as dolphins swim by and vocalize, we record those noises for future analysis. The idea behind using passive acoustic monitors is that they're not subject to environmental or logistic constraints. So compared to boat-based studies, which are limited by weather or daylight, these devices can be left to be recording 24 hours a day, regardless of weather or daytime conditions. However, they do have limiting factors, being memory and battery, which is one of the reasons why we wanted to look into some of the methodological considerations to maximize the data out of each instrument. My study species is the common bottlenose dolphin of Namibia. This is a small resident population, estimated to have less than 100 individuals in the population. They have a very coastal habitat and are rarely seen in depths over 15 meters, with a home range of about 800 kilometers from Lüderitz in the south of Namibia to Moe Bay in the north. And I'm working with the Namibian Dolphin Project, which has been around for over 10 years. This project has a large photo ID catalog, and so we have strong foundational knowledge of the individuals in the population, as well as a catalog of over 50 signature whistle types already identified. So mark-recapture models are based on the generation of capture histories of individuals. And in this project, we'll be basing those off of acoustic captures of signature whistles. So we're not actually capturing the dolphin, but we will be recording their signature whistles as they swim by the recording devices and use those to make the capture histories. With a limited number of hydrophones available and a large range to survey, it's important to consider how you deploy and where you deploy these devices in order to maximize your acoustic detections with the resources you have available. So for this study, we used 3 hydrophones, which we deployed in an L-shaped array. You can see from the red dots on the map. And we're looking at 2 questions in particular. One was the array size and configuration. So using those 3 hydrophones, we came up with 7 array configurations designated by their cardinal codes. So the 3 singular arrays and then a combination thereof, with the last one being a combination of all 3 hydrophones to use in our data set. The second question we wanted to look at was duty cycling. So you can set recording schedules on these devices to turn on and off, thereby increasing the length of deployment. However, you do, by turning off the device, you are not recording for certain periods of time. So we wanted to look at what the most effective way to set a duty cycle was in which the results were still maximized. So we've recorded 16 days on the 3 hydrophones, and we're able to identify 1,099 signature whistles assigned to 21 individual signature whistle types or assumed individuals. One thing I want to point out here is that given the close proximity of the hydrophones, the amount of time that the dolphins were around each hydrophone was basically equal, somewhere around 25 hours over the course of the 16 days. However, there was still a difference in the number of signature whistles identified at each hydrophone. But even with these similar encounter durations at each hydrophone, we did see a difference in the capture probability based on the array configuration. To test this, I used multiple models, mark-recapture models, of which I'll be presenting the Cormach-Rayleigh-Saber model with a constant survival and constant capture probability, as this had the lowest AIC. And again, the capture probability is the parameter of interest here. So in this graph, we can see capture probability is on the y-axis, and then each of the 7 bars represents one of the array configurations. On the left side, we have the 3 individual hydrophone arrays, so the northwest, southeast, and southwest. And we can see that the northwest hydrophone on the far left estimated a capture probability much higher than the southwest array. However, what is more interesting to note is that with the addition of more hydrophones into the array, so with the double hydrophone configurations as well as the triple hydrophone configuration on the far right, the estimated capture probability did not increase very much. So because of this, these results show that array configurations do affect the capture probability. However, we believe that the location of the hydrophone is more important than the number of hydrophones you have. As for the results of the duty cycling, I again tested this using the Cormach-Rayleigh-Saber model with constant survival and constant capture probability, and the capture probability parameter was the parameter of interest. I tested this on 2 different array configurations, the northwest array and then the triple hydrophone array configuration, using 5 different duty cycles, 3 at 50% and a 1/3 cycle and a 2/3 cycle. I'll be focusing on the triple hydrophone array configuration as these results were the clearest. What we can see from this chart, that the full cycle clearly did the best and that each of the subsequent duty cycles had a lower capture probability, which was to be expected. However, the 5-minute on, 5-minute off cycle performed better than the 30-minute on, 30-minute off cycle. So these are both 50% cycles. One was just shorter than the other. So what we can see here is that duty cycling does extend deployments, but it does lower capture probability, as within the day you are lowering the amount of recording time. But we believe that with temporarily sporadic vocalizations, such as signature whistles, shorter duty cycles are more effective at capturing these vocalizations. So in conclusion, we believe that it's important to have background knowledge on your species and signal of interest, which you are attempting to record. So with bottlenose dolphins and their signature whistles, they're fast-moving animals, and these whistles are produced very sporadically. So consider a contrast with humpback whales. If you're attempting to record their song, the animals are stationary and the song can go on repetitively for hours and hours and hours. So for, say, with duty cycling, you can have a much longer duty cycle and still guarantee that you're going to record the song. Whereas with signature whistles and bottlenose dolphins, you want to deploy the hydrophones in an area where you are most likely to acoustically capture them. Moving forward, this method of mark-recapture with acoustic signals, you're dependent on the animal producing this signal, and there are a lot of behavioral or environmental factors that go into the animal producing these signals, such as group size, composition. So we want to investigate further those behavioral and environmental pressures which affect the vocal production of signature whistles, and then also move these capture histories into various other models, such as spatial capture-recapture or social network analysis. And one thing we're really excited about is that this method could potentially be transferable to any other species that has a unique individual acoustic signal, both terrestrial or marine. So we hope that the study and the work that we're doing here can help studies across the world of bioacoustics. And with that, I would like to thank you for listening to my presentation, the ABC community for putting on this conference and making it all happen still, as well as Stellenbosch University, and then, of course, my funders. Thanks so much, and I am available for any questions you have right now.